you can actually just watch the dynamics of the system, right? So what we've got is the microbes and the viruses at regular concentrations and the DOM here. And what they do, of course, is that initially the viruses drop off, right? But the bacteria will be growing, and then the viruses catch them, and they go through a birth cycle, and then you end up with this, they eventually reach this steady state where you're going to have like 10 to the 7th viruses per mil, 10 to the 6th bacteria per mil. Okay. However, in the places where you don't have the viruses, where you have very few of them to begin with, they really just degrade and they never come back, so you never see them. And in these cases, the bacteria will grow up to 10 to the 7th per mil because they're not being predated down. Okay? And of course, there would be more energy used by the system here than there would be this system here, right? Okay. All right, now if you do the same experiment, yes?
So what about induction events, like within the microbes?
Yeah, so it doesn't happen in the marine stuff. So the question would be, do the lysogens, why do the lysogens not just induce enough that we can get the viral communities back? For some reason, the marine system, it doesn't happen, right? We don't see enough production of temperate viruses to replace that lytic community that was there originally.
So that means they're following a totally different strategy. It's not like they're just hanging out waiting for opportunity or anything.
Right, it may be that, yeah, and maybe they're not set up because of like how often, it could go back to your guys' original question. Maybe when they do an attack, right, they go through a lytic cycle and then they do an attack, maybe they all become lysogens, right, as opposed to entering a lytic cycle, right? But that's actually pretty repeatable. Like we've done this experiment in a whole bunch of number of ways. That's actually Victoria has been doing it recently, and she gets basically the same answer. All right, so this experiment is the same thing. The only difference here is we're going to change where the viral community comes from. All right? So what we're going to do is we're going to have the microbial community is going to be from seawater, and we're going to be adding it to just dissolved organic matter from seawater so that they can grow. Okay? And then we're going to add a viral community that's from somewhere else and ask, can it grow in the seawater? Okay? And what you can see, except for this one here, that basically in soil, freshwater, and sediment communities all give you a viral community that does the same behavior on the marine environment. Right? So this means that the viruses that are in Lake Murray could flow down the river and actually probably infect marine microbes downstream of it. Okay? So this means that you can be moving viruses around the world through different biomes. Okay? All right, and so then that, of course, leads to this part of it, which is essentially everything is everywhere question. And the idea of these sorts of models are that really microbes can get around so much that it doesn't, you don't actually see local adaptation. They just get around and they get selected up in a particular environment. And we tried to test this, which this is Florent's paper. So we got all these samples. These are mixture samples, just so you know. So this is from the Arctic, where we've got a whole bunch of different sites and a whole bunch of samples all thrown together because sequencing was so expensive at the time. This is Curtis Settle. This is Curtis Settle gave us this one from British Columbia. This is also Curtis Settle. Yeah. And then this is Craig Carlson gave us this concentrate out there. And we just sequenced them, okay, using the same methods that you know. In this case, it was actually the genome feed, and that becomes important, right, because that introduces a bias. And then this was one of these first pyrosequencing studies. So these will only be 100 days. And what we found is that, one, that we had a whole bunch of these single-stranded microphages. And this was really the first time that anybody had seen these in the environment. And what we think that the reason is that we got them is none of our methods before would have actually cultured them or gotten them to show up in the metagenomic libraries. It's also that they're overrepresented. We know that now because that genome feed replicates that single-stranded stuff better than it does the rest of the system. So there's a bias in here. All right. The other thing that we saw is that you actually see more temperate-like viruses, so things are known to form prophages in the Arctic than the other parts of the system, in the other environments. So maybe because of that burst and bust cycle that you have going in the Arctic, maybe being a temperate phage makes more sense, and maybe if you did those experiments that we talked about earlier, you would see a lot more temperate behavior. All right, and then this is probably the world's first recruitment plot. I'm pretty sure it is actually. And what we're doing here is we're going to take these samples, and we had the Prochlorococcus one, that phage, that is about the same size that Matt Sullivan had sequenced, okay? And then this is Roseophage along here, and then we're asking where did the fragments recruit to across it. And as you can see, Prochlorococcus, the phage, was mostly only in the Sargasso Sea, which makes sense because that's where we isolated that phage from. Okay? And then we see almost no overlap in these kind of nearshore environments, let's call it. And you can see the only things that we're really seeing are, these are where you have an active site in an enzyme and you get recruitment to it. Right? And then the Roseophage is basically in these nearshore environments, right? Which is, again, makes sense because that's where it was been. And so recruitment plots are still one of my favorite things. This, with this other analysis, this is probably Unifrac from way, way back. And what this is probably, if I remember right, we're looking at well, I know we're looking at geographical distance between versus phylogenetic distance. And what we basically saw in this case is that there is some signal of the geography going across from the equator, well, the Sargasso Sea up to the Arctic, which you can see here. Okay. Right. And then when you do the facts analysis, so what we're going to do here, remember this is where we look for the overlaps and we ask what diversity is. So if you do that, you get this interesting result. So this is the number of predicted genotypes in the Arctic, the British Columbia, Gulf of Mexico, Sargasso Sea. Okay? And in the British Columbia, for example, you see like maybe over 100,000 predicted viral genotypes. Okay? Here in the Arctic, it's much lower. Then it's going to be somewhere around 10,000 for these other sites. You can't see that now. That would say 1,500 or 15,000 here. Okay. But what's interesting is that if you just take all of them and you put them together and you say what does facts give you, it only gives you about 58,000 genotypes, which is lower than what we got here. Okay. So the only explanation is that there's something that's compressing that, right? That there's a sharing going on. And this is what led to Maxify, was this observation. All right, and so this is, Florian gets credit, but actually the person that figured all this out is actually Ben Feltz, because he sat down and ran a Monte Carlo. And basically how this thing works is that you mix the communities and you look for those cross-contigs, right? And you're asking this question. So this is my rank abundance curve, right? Did it change when I change it, compare it against one sample and another sample, right? Did the rank abundance curve look the same? Okay. Or did the relative ranks change? So we have everything is there, but they just changed their relative abundances, right? So your most abundant thing is down here now. Got that? Or you could just have new genotypes coming in, right? So this is called percent permuted, and this is percent shared. And that's what we're always plotting on those things. All right, and this is just the answer. So if you do that for all of those samples and you ask, what we really find is that across the oceans, 100% of the genotypes are shared, okay? But the relative ranks of the most abundant ones have changed a lot. Okay? So this means everything is everywhere from the viral point of view, okay? But their just relative importance is changing.
So this is like standard rare biosphere stuff, right?
Say it
This
again.
is like standard rare biosphere.
Oh, fuck the rare biosphere. That's just an
I
artifact.
think you just
Yeah,
proved it, right?
Yeah. No, I mean, this is, yes, but we've always known that there's like basically a
tail.
seed bank or something way the fuck out there, right? That, yeah. Don't worry about the rare biosphere, just gives me a headache.
Does
Right.
it allow for any mismatches? Do you say like non-mismatches?
No, this doesn't. In the initial one, it was 98% identity over 25 base pairs, and now it's 100% identity over 35, I think is what the assembly is. Yeah, how's that? So these are exact matches in this case. So this one may be using the old criteria though. But that was below our error rate. Right? Our error rate of the 454 at the time was higher than what we were allowing in this one. All right, so where does this leave us as a lab? So one, we spent a lot of time from let's say 2000 to 2006 making sure that our library